process. We're right on track. Last process for tonight, transduction. Okay, so transduction is our final process. I saved the best for last. It's so fun. Okay, why? Because it's the first time this semester we get to look at viruses. One of my favorite. Yes, I know, one of my favorite. We'll have a whole week of them later on in November, but a little snippet tonight. So, this process of transduction is the process of horizontal gene transfer that involves viruses. We call these viruses bacteriophage. Who do you think they infect? Bacteria. Okay, so they're called bacteriophage, a special virus that infects bacteria. We'll also be working with them in lab later on. Okay, so transduction, last process of horizontal gene transfer, number 3, transduction. Okay, so transduction involves bacteriophage. Okay, and these are viruses that infect bacteria. Okay, what overall do they do? And then we'll go through step by step. But overall, these bacteriophage act as little shuttles. They're going to take pieces of DNA from one bacterium and bring them to another bacterium. So they're acting as kind of an intermediary. So these bacteriophage will transfer DNA from one bacterium to another. Okay, up here I have a poor little picture of E. coli. He's just being bombarded by hundreds of bacteriophage. Feel sad for him? I'm not sure now. All right, so transduction. So the cool part about this is we get to look a little bit at viral replication because we need that to understand transduction. So it's kind of a little preview into virology. Okay, so here's the idea. Bacteriophage are a special type of virus. The way they work is they land on their bacterial cell, okay, and they inject their viral DNA into their bacterium. We would call that usually a host cell, bacterium, okay? So I'm going to look at this as a stepwise. So transduction steps. We'll go through in this process viral replication as it relates to transduction. So 1, the bacteriophage attaches to a host bacterium. Okay? And again, as I mentioned, once it attaches, it injects its viral DNA into the bacterium. So the phage, I'm just going to say for short, injects the viral DNA into the bacterium. Okay? Here to me is one of the coolest parts. When it injects that viral DNA, which is shown in red, I know it's cut off up here, but it's red, and we have the circular chromosome in blue of the bacterium. Okay? When it injects its viral DNA, okay, the cell treats that DNA as it would any other DNA. So the cell becomes kind of a slave, right? It takes the viral DNA, it transcribes it, and it translates it, just like we talked about. Okay, guess what? This is, again, one of the coolest parts to me. On that viral DNA, after it's transcribed and translated by the host cell, it actually codes for proteins that go and chop up the host cell DNA. Oh, that's cool, right? Why is that cool? Because, hey, now it's kind of diverting all the cell's resources and energy to its own viral DNA. It's like, oh, let's chop up the host DNA because you're not going to need that anymore. Okay? Yeah. So the phage injects viral DNA into the bacterium. Again, this viral DNA codes for proteins that degrade, is the term, break down, the host cell, that's the bacterium, DNA. Make sense? Okay, and again, there's a benefit to the virus because basically with the host cell DNA all chopped up, it's really not going to mess with its own DNA fragments. But it'll still go ahead and transcribe and translate the viral DNA, and it'll replicate the viral DNA, okay? And so the host cell does all this work, basically, of making new viral parts using the viral DNA. That makes sense? No, cool, right? All of these new viral parts are made, so more viral DNA, more viral proteins, okay? After all of the parts are made, then we go to what's called assembly, where the viral proteins are stuffed with viral DNA. It's kind of a key part. So let me recap this, make sure we get this part. Okay? So number 3, after the host cell DNA is degraded, okay, the host cell uses the viral DNA to two things: make viral proteins, we know all about that, right, by transcription and translation of the viral DNA. Okay, also it makes more viral DNA through DNA replication. Okay, does that make sense? So again, it's made all the viral parts now, just using the instructions that were injected by the virus, the viral DNA. Okay, after these parts are made, then we have assembly of viral DNA into viral proteins. And again, this happens inside of the host cell. So again, this is a key point. With these VAGs, and we'll see more about their structure when we get to virology, but as an intro, they have these heads, we call them, that are stuffed with their viral DNA. That's what we mean by assembly. So the viral proteins make up these heads, and then we stuff them with viral DNA. Okay, that's assembly. Okay, why is this a key point? Well, guess what? Most of the time, because we have lots of copies now of viral DNA around, right? Most of the time, and it's hard to see color-wise, but they're showing 1, 2, 3, 4. Okay, most of the time, 4 out of 5 here are stuffed with viral DNA. But remember, we have pieces of bacterial DNA lying around, right? So occasionally, okay, one of these viral protein heads is stuffed with bacterial DNA. We give that a special name. We call it a transducing particle. Okay, so it's the viral protein head that now contains bacterial DNA instead of viral DNA. Cool? This part's one of my favorites. So, in this assembly, okay, I'm going to blow up the assembly because again, this is important. This is where we actually generate the particle that will be responsible for horizontal gene transfer. So 4 continued, the assembly. In the assembly, the viral protein heads are loaded, we say, with viral DNA. Okay? Sometimes, though, okay, over here, sometimes, not very often, a viral head is loaded with a piece of bacterial DNA from the host. Okay, that's host cell DNA, right? We call this a transducing particle. Okay, that's the hardest part. Okay, the hardest part of the cycle is done. So again, the host cell is infected with this viral DNA. Okay, it uses the viral DNA to make all new viral proteins, viral DNA. Most of the new viral particles consist of viral DNA. Occasionally, though, we get a viral particle that has bacterial DNA in it. This is a special one. We call it a transducing particle. Okay? After all of the viruses are put together, whether or not they contain viral or bacterial DNA, they are all released. So the viruses are all released. So sad, we'll see it later, but they burst the host cell, they kill it. Okay? So they're all released. We'll come back to that later. What happens here though? All of these viruses, right, then are free to come along and infect new host bacteria. This is the part finally that relates now to gene transfer, now that we had some viral replication, right? What we really care about for our talk here on horizontal gene transfer is the transducing particle. Okay, all the viruses, you know, with viral DNA, they're going to go through the same process, most likely, you know, just killing their cells, their host cells, right? Through the whole process we talked about. However, the transducing particles, they come along, they infect a new host cell, just like any virus would. However, what are they injecting? They're injecting not viral DNA, but Bacterial DNA. Here's finally the horizontal gene transfer, right? So this cell receives that piece of bacterial DNA. Think of this analogous to what we just looked at with transformation. It can use that free piece of genomic DNA and integrate it into its chromosome. We would call that a stable transduction or a stable gene transfer, they have labeled here. Okay? It may also degrade it like we saw with transformation. That would be unstable or unsuccessful. Okay? But again, the point is that these transducing particles have the ability to create genetic variation, and I'm creating recombinant DNA. Okay? By injecting the host cell with bacterial DNA instead of viral DNA. Okay? So here what we care about are the, I'm going to say TP, transducing particle, okay, can inject bacterial DNA into a new host bacterium. Okay? And again, just like we saw with transformation, if it is integrated into the host chromosome, okay, that would be a stable gene transfer, stable transduction, either one is fine. Specifically, it's a transduction, right? Okay. And again, this also results in recombinant DNA. Pretty sweet, right? So again, viruses naturally can do this in the environment, where very seldomly they'll get that piece of bacterial DNA loaded in a viral head, and they will be able to transfer that to a bacterium that can then incorporate it into its chromosome. Again, the converse is if the bacterium that receives that DNA doesn't use it, or it degrades it, these would not be successful. The organism wouldn't be able to use that piece of DNA to make new proteins. Okay?